Hi, my name is Jacob Curtis, and this is my presentation on the TERC TERC gene. Let's break down the name. TERC stands for telomerase RNA component. It encodes the piece of non-coding RNA used in a larger protein called telomerase. We'll talk about what that protein does later, and all you need to know for now is it extends telomeres. I hope you remember what telomeres are. If not, we'll crash course that later. The specific piece of RNA that is coded is called TERC for shorthand, just like the gene, but it's also referred to as HTR, and I will refer to it as such just for the rest of the presentation, just for clarity distinctions. Again, we'll be calling the gene TERC and the piece of RNA HTR. Location, size, and species. Telomerase and TERC occur in lots of eukaryotes, from yeast to fruit flies and all the way to humans. We'll be focusing on how, TERC presents itself in humans for this presentation. TERC in humans is located on the minus strand of chromosome 3, starting at base pair 169,764,462, with a total length of 599 base pairs. The genes that encode other parts of telomerase, again, the largest structure in which HTR appears, are all on other chromosomes. HTR is 451 nucleotides in length. Interesting tidbit, in yeast, it's about 1,200 nucleotides in length. Kind of crazy. After being transcribed, the RNA immediately starts forming and folding on itself. No splicing, nothing. HTR has three major parts that you can identify, or that we can identify. The core domain, that part in red there on the diagram, is what holds the part of exposed nucleotides that will be used to base pair to extend and make more telomeres. The human telomere is a repeat of TTAGGG and just loops that for its whole length. If you look at the lettered part of the core, you can see the pattern for the base pairing, AAUCCC. The extra part is to help in reverse transcription. We'll get to what that means later in the presentation. The other parts are pretty much structural. CR4/CR5 binds to the TERT, telomerase reverse transcriptase, another subunit of telomerase. The core domain also binds to TERT, but that's not its main purpose. And the last, HACA, helps HTR bind to telomerase as a whole, so the whole structure. So here's an actual 3D model of what telomerase looks like. That green part is that subunit I mentioned earlier, TERT, telomerase reverse transcriptase. It's what catalyzes reverse transcription. And see that yellow bit that looks like DNA sticking out the bottom? That's the HTR, the telomerase RNA component. The TRC, that's our guy. The exposed part is the AAUCC we saw earlier, and a little bit of that those extra exposed nucleotides. That's what binds the telomeres. The other parts, like discrin, NOP10, all just help identify and find the telomeres that the telomerase wants to bind to. All right, reverse transcription. This term I've been throwing around. You want to know what it means? First, though, the aforementioned promised crash course. So every time a cell divides, it needs to copy all its DNA, its chromosomes. The process through which this is done, DNA synthesis, isn't perfect. So every time a chromosome gets copied, a little bit of the end gets cut off thanks to lagging strand synthesis. Now, we don't want to roll the dice and just hope that lost information wasn't important. So chromosomes have built-in ends of useless repeating nucleotides that don't do anything. These ends are called telomeres. And when DNA is replicated, they cut off a little bit, but no genetic information is lost. But wait, what happens when the telomeres run out? I hear you ponder, you inquisitive listener, you. Well, that's when the cell enters senescence. It stops replicating and it eventually dies. Forever. Reverse transcription, though. What could this complicated process be? It's just regular transcription, but the DNA is copied from RNA. You will remember during regular transcription, a piece of RNA is copied from a strand of template DNA. Then it goes on as non-coding RNA or gets to be translated into amino acids. However, in this case, HTR is attached to the telomere with a couple of nucleotides, those ones that we pointed out earlier, as you can see in this thing, starts out as CAAUC. Those are the exposed bits we saw before. It then and it starts synthesizing DNA. This is actually done specifically by the TERT part of telomerase, but HTR is the template of which it is built off of. It then detaches and just rinse and repeats till a suitable length of telomere has been synthesized. DNA polymerase does the work copying the lagging strand from the extended template strand the telomere just built. And bam, now you know reverse transcriptase. Onto the alleles. The TERC gene is inherited autosomally from parents to children. I found 13 alleles for the gene with about 5 expressed phenotypes. They are dyskeratosis congenita, a type of inherited bone marrow failure, generic bone marrow failure, pulmonary fibrosis and bone marrow failure, and finally wild type, just functional. If you haven't noticed yet, you can make 2 categories for all the phenotypes: healthy expressions and horrible disease. I haven't included photos of the disease for the sake of viewers' discretion, but I can't stress enough that they are very deadly diseases / symptoms, and those who survive them do so only by the grace of modern medicine. And I'll go into detail why they are bad. Bone marrow, the stuff inside your actual bones, is host to stem cells. Stem cells are cells that can transform into other differentiated cells. What kind of cells specifically? Technically, any other cell in the human body. Okay, so that's not specific. The ones in your bone marrow specifically turn into red blood cells and white blood cells. Red blood cells that transport all the oxygen throughout your body, the oxygen your body runs on, and basically makes the whole vascular system work. And white blood cells, the primary cells that our immune system uses to fight infectious disease. 2 very important cells, backbones of the whole human apparatus, really. Stem cells also have active telomerase in them, which differentiates them from regular senescent cells. And so the fact that telomerase exists in these cells, extends their telomeres and pretty much extends the amount of time that these stem cells can divide, essentially limitlessly. So when stem cells fail, bone marrow fails, and when bone marrow fails, you stop producing red blood cells, white blood cells, platelets, and more. You're left up a certainly aptly named creep without a paddle. Again, I'm not gonna show visibly what the symptoms are. You can look those up yourself if you're curious. Now, what are the details of these alleles that cause such a fish? Well, 9 out of 13 of them are single nucleotide point mutations. For allele number 2, the swapping of cyanine to guanine at base pair 408 leads to dyskeratosis congenita. G2A at BP58 is bone marrow failure. C to T at 116 is bone marrow failure again. So on and so on. So it really goes to show just a single nucleotide off can really ruin the whole shebang. Alleles 1, 6, and 11 all involve base pair deletions and similarly lead to the previous phenotypes of bone marrow failure, pulmonary fibrosis, and dyskeratosis congenita. And that is Finn. Here are my sources, and I hope you've had a good time listening. Feel free to email or ask any questions in person. I can't promise a good answer, but I can promise my time. Thank you for listening.